Hello, everybody. Today I'm going to talk about two techniques regarding neutral lipid sustaining, BODIPY and quantum dots. BODIPY is a highly fluorescent chemical compound that has this structure here and this huge name, which I'm not going to try reading it, and tends to be strongly UV absorbing of small molecules, emitting relatively sharp fluorescence peaks with high quantum yields. The main characteristics of BODIPY is it's a highly lipophilic structure. It's a neutral fluorophore, and it is used to label high range of lipids such as fatty acids, phospholipids, cholesterol, esters, and ceramides. Also, BODIPY dye is independent of chemical environment like pH or polarity. This is an overview of the BODIPY technique workflow, which is very simple and can be coupled with other techniques, such as FACS in this example here, but especially with microscopy or immunofluorescence assays. Usually, BODIPY dye is solubilized in PBS, and samples are incubated with BODIPY solution in the dark for 30 minutes. Depending on the coupled technique used after BODIPY, it would be necessary to fixate the staining. Here, it's a quick video showing a comparison of a microscope view of lipid droplets labeled with BODIPY and other type of staining. Well, BODIPY applications it can be applied in several technological fields, such as organic lasers, probes, and sensors, such as imaging diagnosis, cancer, photodynamic therapy, and also in light collectors and photovoltaic devices. Well, BODIPY has many advantages, such as it has a higher fluorescence quantum yield. It has a stable absorption in water. It has a great photochemical stability, wide range applications, and is also stable in physiological conditions. The only limitation of BODIPY is it doesn't work very well for antioxidants evaluation in lipid peroxidation. Well, now talking about quantum dots. Quantum dots are man-made no-scale crystals that can transport electrons. When UV light hits the semiconducting nanoparticles, they can emit light of different colors. Different type of quantum dots can be excited with the same light wavelength, and their narrow emission bands can be detected simultaneously for multiple assays. The fluorescence bands of quantum dots depend on their composition, size, and shell thickness. Well, quantum dots can be used in biological research for studying kinetic processes in cells, such as transport mechanisms, diffusion movements of membrane, and also to study functional heterogeneity of cells. In medicine, quantum dots are used for contrasting and molecular diagnosis. Well, quantum dots also has a few advantages, such as it has a high photostability, resistance to photo bleaching, being 100 to 1,000 times higher than organic fluorophores. Also, it has a high quantum yield and a long lifetime. Also, it has a wide absorption spectrum, and it is resistant to chemical and biological degradation. However, quantum dots has a few challenges and limitations as well. So it has incomplete elimination after injection into an organism. It has a possibility of non-specific binding with any organic molecules. And it has a high toxicity, especially because of its incomplete elimination. And it has to be careful when using in vivo systems. Also, it has instability and increased hydrodynamic diameter after interaction with the serum proteins. Well, this is an example of BODIPY. So lipid droplets are organelles with major functions in lipid metabolism, energy, homeostasis, and intracellular transport, and have multiple roles in infections and inflammation. So this paper here describes that monocytes from SARS-CoV-2 patients have an increased lipid droplets accumulation compared to negative donors. Well, they demonstrate the essential role of lipid metabolic reprogramming and lipid droplets formation in SARS-CoV-2 replication and pathogenesis, opening new opportunities for therapeutic strategies against COVID-19. Well, in this situation here, BODIPY was used to label lipid droplets, which were analyzed by immunofluorescence assay. And last, this is an example of quantum dots. So labeling viruses with fluorescent materials is the prerequisite for exploring virus cell interactions. Compared with organic dyes and fluorescent proteins, quantum dots are an excellent for long-term single particle tracking due to their unique photostability and high brightness. So in this work here, taking pseudoribus virus as a model, a mild strategy to efficiently label viral envelope lipids with quantum dots is proposed by harnessing the biotinylated lipids, self-inserted cellular membranes to monitor virus cell interactions in real time. And those are the references we used, and thank you very much for watching.